You can start. Good. So welcome everyone. I have to apologize that I cannot be at Las Palmas. I would have loved to do the course, but I got an injury on my ankle, which makes me a bit immobile at the moment. But it's getting better already, so there is hope that I will be mobile again soon. Okay, I will talk about using the RedoxSensorGreen to estimate single cell respiration. I'm at the Department of Functional and Evolutionary Ecology at the University of Vienna and have a joint appointment with the NIOZ in the Netherlands. So in contrast to what Morten said, I will not talk about pressure effects because we don't have really measurements on the role of pressure on respiration. I mean, we have done now some laboratory experiments, but this is really too premature to present. But so the question is, I mean, why do we want to measure single cell respiration when we have all these fancy methods around which have been presented this morning with using Winkler, INT and everything, all the other techniques and optodes and so on. The idea is that we would like to have an indication of single cell respiration is because we have complex microbial communities. We have different populations with different life strategies. Morten has shown some impressive slides on microbes colonizing particles and staying on particles, but we have also free-living microbes. We have microbes adapted to different nutrient conditions, to oligotrophic nutrient conditions, copiotrophic nutrient conditions. So we have also microbes adapted to utilize different substrates and so on. So it does make sense to get to look closer into the community. And this will allow us to get a better mechanistic understanding on how microbial communities are organized under different conditions and so on. And so we used this RedoxSensorGreen, which is converted in active cells, actively respiring cells, to a green fluorescent product, which can be detected. It can be detected easily with an epifluorescent microscope using an image analysis system, or by flow cytometry. And with using flow cytometry and cell sorting, you can sort, for example, these cells which are fluorescently labeled, and you can do, for example, sequencing afterwards to get information on who is active to what extent. The manufacturers, which is in Vitrogen, the recommendation is that the staining, one should use 1 micromolar of RedoxSensorGreen, and this would stain about 10 to the 5th to 10 to the 8th cells per milliliter. And actually, this is pretty much independent of the sample volume. And I mean, using RedoxSensorGreen, it has an advantage over, for example, CTC, which is also a tetrazolium salt-based fluorescent substrate used in the past for visualizing essentially respiring cells. But RSG is superior because it has higher spectral properties, fluorescent properties, and this allows shorter incubation times. And also, and this is sort of also important, it has no known toxic effects. So we used that, and then we used that for natural communities. We were first determining, I mean, the optimal incubation time, as you can see on the left panel here. using coastal seawater. And so what we found is that actually after like 30 minutes, the cells were pretty much saturated, or I mean, we have the positive cells, RSG positive cells were all essentially stained after like 30 minutes. So therefore we used 30 minutes as a routine incubation time. And originally we started off with 50 ml sample and 1 micromolar final concentration, as shown here at this panel here, to determine the incubation time. And now we are routinely using for natural communities just 1 milliliter of a sample and 1 microliter of RedoxSensorGreen, which is basically also 1 micromole solution. Because the RedoxSensorGreen, the major disadvantage of that is that it's pretty expensive. So you would like to use as most efficiently this RedoxSensorGreen as possible. And 30-minute incubation times turned to be really enough. And then this incubation can be terminated by freezing, putting them in liquid nitrogen for flow cytometry, and then storing it as -80. The fluorescence is stable for quite some time, actually. Or for epifluorescent microscopy, you simply store it or add formaldehyde, like 2% formaldehyde will do. For enumerating with flow cytometry, you use an excitation of 488 nanometer with a bandwidth of like 525 nanometer emission and a bandwidth of 35 nanometer. This is what we used. And under epifluorescent microscope, you use basically like the FITC block, which has been commonly used and is common in the epifluorescent microscopes around, and the excitation emission is 595 to an excitation, and emission 519 nanometer. And then you can also use like a double staining or triple staining. Even we'll show you later on some pictures on that. You can also double stain it with DAPI, because the excitation emission spectrum does not overlap, and you can also use then oligonucleotide probes, fluorescently labeled oligonucleotide probes, for example. We calibrated essentially this the fluorescent intensity first with some different bacterial strains, as indicated on the left-hand panel, using all kinds of different strains and also different temperatures, like 14 and 22 degrees centigrade, and measuring the fluorescence basically on the x-axis, and on the y-axis we also measured the respiration rates using Winkler titration or and optodes. And so over quite a range of like 3-4 orders of magnitude, we did get quite a linear exponential relationship here, because this is a log scale here. So this was quite convincing, using these different bacterial strains. And then we also went into the ocean and sampled Gulf of Mexico, the open Atlantic of, here it says Mallorca, but it should be, of course, the Mediterranean Sea of Mallorca. And so we use open ocean and coastal waters, and getting again quite a range of respiration rates as determined by Winkler, and measuring at the same time RedoxSensorGreen, the fluorescent intensity. And so what you can do really is, when you measure this Winkler, the rate, what you get. You can break this down to the fluorescence, and you can calculate essentially then the RedoxSensorGreen oxygen consumption, and you can break this down to individual cells, of course. The detection limit there for on an individual cell level is for attomole. Attomole is 10 to the minus 18 mole oxygen per cell per hour. And the lowest cell abundance we quantified is 10 to the 6 cells per liter, so which means 10 to the third per milliliter. So this gives a theoretical detection limit of around 4 picomole oxygen per liter an hour, which is much more sensitive than, for example, conventional Winkler or optote sensors. So basically, this allows respiration estimates on quite a low abundance of populations, and also fairly low activity on a cellular base. The left-hand panel here shows you the gating window in the flow cytometry on live samples, and on the right-hand panel as a control heat-killed community. And you see is heat killing before you add the RedoxSensorGreen really kills the microbes, so there are no respiring microbes, while on the left-hand side you see these respiring microbes here. Okay, and you can also, because these short incubation times of like 30 minutes only, allow you to really look into microbial activities during day and night, whether there are differences and so on, with subsequent cell sorting and sequencing. And this is shown here. Based on the fluorescent signal, the estimated diameter based on the side scatter in the fluorescence, and here the respiration rates of different microbial communities. So like a few archaeal we also got in there, but mostly bacteria, alpha proteobacteria and gamma proteobacteria are sort of dominating. Bacteroidetes in this light green dots. So what you can see is that alpha proteobacteria are respiring quite a lot, and also the gamma proteobacteria, the brown dots, they're respiring quite a lot. And there are some subtle differences between day and night. And these were coastal waters, surface waters. We don't have any data on deep waters at the moment, but this will be done in the upcoming cruise, where we take RedoxSensorGreen and we will incubate it in the ISME and measure at the same time also bacterial production at pressure conditions. This slide shows you the RedoxSensorGreen in a degradation experiment where we had jellyfish detritus and followed microbial degradation. And here on the left-hand panel, we have a triple staining with the TAPI on one hand to enumerate all the microbial cells, then the fluorescently labeled cells with Psi3, which has a nice emission, also not overlapping, and then RedoxSensorGreen. And what you see here, this Alteromonas oligonucleotide probe, Alteromonas specific nucleotide probe, and RedoxSensorGreen with these brightish, shining, actively respiring cells. Also here, this on the right-hand panel, this greenish fluorescent, these respiring bacterial cells. So you can use all kinds of different staining techniques. You can use oligonucleotide probes along with DAPI and RedoxSensorGreen, and you can do with flow cytometry and cell sorting. You can do sequencing afterwards to enumerate essentially, or basically get phylogenetic information on the respiring communities. And you can break down when you do at the same time with the fluorescent staining, when you do like optote oxygen measurements, you can basically take the sum of all the fluorescence in a sample with and relate this to the number of or to the respiration rate, and you can break this down to the number of active cells. Each cell will have a specific fluorescent signal as determined by image analysis, and you can use this relative fluorescent unit and convert it to oxygen. Something similar we have done also with catalyzed reporter deposition fluorescent in situ hybridization, which is basically doing pretty much the same thing, measuring biomass production or uptake essentially of leucine of heterotrophic bacteria. And when you do autoradiography with on these samples, then the halo around, the silver green halo around the cell is related to their activity, to their uptake activity. And also from that approach, you can get deep information on the uptake activity and the biomass production of an individual cell. And now we have also a tool where we can measure the respiration or estimate the respiration rate of individual cells. And this is what, for example, when you break this then down to individual groups and relate this, for example, to the biovolume. For example, here Pelagibacter, which is commonly known as the SAR11, in one of the most abundant bacterial clades in the surface waters of the ocean, then you see that these most abundant groups, they have a really low respiration rate. And while other bacteria are respiring like 2-3 orders of magnitude more, but they are less abundant. So what we generally found sort of as a trend in this analysis, using more coastal waters down to 200-meter depth. So thus far we were working only in the euphotic layer and in the uppermost mesopelagic. We found sort of an inverse relation between the abundance and the respiration rate per cell. So low abundance microbes are apparently mostly highly active, while the ones which are highly abundant, they show lower metabolic activity. And this, of course, needs further verification. And I'm really curious of how this pattern looks like when we do these measurements in the lower mesopelagic and trying to push it to also bathypelagic and under in situ pressure conditions, measuring the respiration rates. What it turns out from these measurements, from this panel, it is quite evident that there is a weak relation between the cell-specific respiration rate and the cell biovolume. So it's not like that the larger the organism, the more respiring, the higher the respiration rate is necessarily. Okay, yeah, and as I mentioned already, if you use flow cytometry, you can easily sort these RedoxSensorGreen positive cells, and you can sequence them. And the nice thing is what I concluded here, and what we concluded, that the most abundant microbes are only marginally important in terms of respiratory activity. This corresponds quite well to what we found using a proteomics approach and the genomics approach. And this shows you some, these columns are epipelagic, mesopelagic, and bathypelagic waters from the Atlantic, where we did metagenome analysis, and basically this gives you the relative abundance of Thermachyota, Urearchaeota, alpha proteobacteria in green, and gamma proteobacteria. And when you see the relative abundance of metagenome, the metagenome level, on the endoproteome level, they are pretty similar, which gives some confidence there. So the endoproteome is also pretty similar to the metagenome, but we were also collecting the so-called exoproteome, which is essentially the contribution of bacteria to the protein fraction of the dissolved organic matter pool in the ocean. And what you see is, well, the metagenome distribution and the endoproteome distribution is fairly similar, with the alpha proteobacteria dominating. Gamma proteobacteria are also important, but they dominate excessively the exoproteome. So the dissolved proteins originating from gamma proteobacteria are by far the most important ones in the DRM pool. And this, of course, one can break this further down, what these gamma proteobacteria are, and this is shown here. So looking and zooming into the gamma proteobacteria, so it's by far essentially, again, very similar, the metagenome level and on the endoproteome level, the two left-hand panels, but very different from the exoproteome level, where clearly Alteromonas is dominating and releasing the most important contribution of the bacteria into the DOM pool, which means that, and Alteromonas are quite well known to have high turnover rate, relatively high respiration rates, and also high loss rates. And this is, I think, the important issue, that fast-growing cells have also high loss rates. They are highly active, but also the loss rates are fairly high. And this has been also found, for example, shown that these highly active cells are also preferentially grazed by flagellates. And of course, these highly active cells, they also have higher loss rates by viral infection. Okay, and I think that's it, what I have to tell you. So RedoxSensorGreen is definitely something which has quite a number of advantages, and it offers really insight into the communities, what is an active, which fraction of the community is more active than another one. So it also offers you insights into a single population. So within a population, probably there are also some more senescent, more juvenile cells. So one can also look really even into specific populations with that. And you can also look, for example, and compare free living versus particle-associated ones. This would also be challenging. So this is definitely, I think, RedoxSensorGreen is definitely something I can recommend to everyone who would like to do single cell respiration estimates. And with that, I thank you. Unfortunately, I cannot demonstrate you this in Las Palmas, but it's, I mean, the application sort of is pretty straightforward, using an epifluorescent microscope and incubating it for a short period of time, and then looking under the microscope, and then you get, with the image analysis system, you get a fluorescent signal of each individual cell, and you sum this up and you relate this to your Winkler optote measurement, and then you can calculate the fluorescent signal and convert the fluorescent signal of each individual cell to a respiration rate. Okay, thank you very much. Are there any questions?